Here is Yogesh Verma from Aalto University, and he's a PhD student. And he'll present his paper on Let Physics Guide Your Protein Flows, Topology-Aware Unfolding and Generation. I'm really looking forward to your talk and discussion.
Yeah, so thank you for the kind introduction. So let's start.
Mhm.
So we know that diffusion and flow models have been extensively applied and revolutionized the field of structural biology, spanning molecular generation, binder design, protein structures, and many more. Prominently in the field of, let's say, protein generation or protein structure generation, most of the generative models follow like an SE3 equivalent or invariant diffusion flow method, where they kind of like learn like a SE3 flow or a diffusion, which they decompose in like learning an SO3 flow, which is on the space of rotation, or an R3 flow in the terms of Cartesian coordinates, which we will discuss more like how they represent it. And they try to learn have a forward process that converts this data, let's say a simple protein structure of having like beta sheets or alpha helix using the SE3 flow to like a random noise in a 3D space, which you can see it. Although it works pretty well, there have been like these diffusion or flow models typically use, let's say, linear noising process to such that the forward kernels can be analytically tractable and easily usable. And they kind of disregard that the protein itself is like have like a certain physical structure, but they treat each residue as independent point, like a point cloud structure, neglecting their physical realism. And empirically, it has been seen in like numerous papers across the literature that it kind of loses the structural integrity, which you can see like, let's say there is a big protein structure, there is a correlation between the subsequent residues, but now they're completely moving into like a certain disconnected soup of residues. And it has been observed empirically, like in many of the papers, they can lead to like disconnected chains, steric clashes. Can they even learn the physics of like this kind of like protein folding, or like how does the switches happen, and many other certain like memorization and those kind of stuff, which is also there. So in order to see this, we try to answer like a following question, like a study a following question. Can we utilize like some, let's say, physics or some kind of thing to devise like a physically motivated noising process rather than this randomly taking to a noise, and use it like in just say generative models for protein design or like protein structure prediction or something. So this can help in also like incorporating physical inductive biases in these kind of models, which are kind of currently kind of missing out on by their neglecting their physical realism. And this also kind of moves these models more towards, let's say, dynamical learning for protein structures, because currently we only learn like say a static structure that is been observed in like an X-ray crystallography or certain kind of thing available in the PDV. But using like a certain physically motivated like a process, it kind of learns how like the internal, like let's say, physical correlation works inside like a protein structure, and try to make it like a much better in learning, let's say, a little bit more robust in like, let's say, in conformational landscape or like a protein structure. Because usually in in nature or like this, the protein exists in like a certain conformational distribution rather than a static structure, which is constantly wiggling around in like a, having like a structure. So this kind of also motivates that kind of thing. So what we try to seek is kind to have like a certain physically motivated, like a forward noising process that kind of unfolds the protein into like a linear chain of protein amino acids. So this comes from like a first principle biological intuition that what can be like a good prior for like it's a protein structure. So one of the like the biology people will say like, okay, it can be just like a linear chain of amino acid connected together that has no information about what structure it can take, but it only depends on like, let's say, like how big the linear chain of amino acid is, or what kind of amino acid is, to tell like how does the protein will fold into like a certain place and have like a protein structure. So we kind of try to formulate it like in a that way process that also like preserve this topological integrity rather than breaking them around, and also avoiding clashes, so that these like residues at the end doesn't go clashing into the residues at the bottom, but we get like a linear chain of amino acids, kind of like a good way, which can also be represented in this diagram. If you move from right to left, you can see like a forward process coming from like, which subsequently like removes like the structural information and make them like a like kind of unfolding them into a linear chain. But before going how we usually parameterize this, let's see how we represent a protein structure in real life. So a protein structure can be represented using an angular representation. So angular representation means like they can be represented in terms of the dihedral angles and the bond angles as shown in the figure. So these are the C alphas for each of the residues, amino acid residues, the carbon, the nitrogen, the oxygen, and the alkaline group. And we represent it via like dihedral angles and the bond angles of theta 1, theta 2, theta 3, and phi, psi, and omega. On the other hand, it can also be represented in the terms of frames, which have been used by AlphaFold and other bolts and other kind of methodologies. So what they try to do, they represent it in a term of like a rotational matrix, such as in the system R, which represents using by a Gram-Schmidt normalization of these vectors corresponding to each of the subsequent residues, and X represents the Cartesian coordinate of the C alpha. And this is how it technically looks like, this kind of representation when you do it. So it kind of tells you like how each residue is relatively oriented and what is the translation component of it. So it kind of disintegrates into decomposing into two terms. Like one is the rotation, like where it is like rotated or aligned well with respect to the origin, and also how far it is located. And there is a bijection between the angular representation and the frame representation, because using the angular you can get Cartesian coordinates of the protein structure back, and from that you can construct the frame representation. So they are interchangeably work via like this neural reconstruction frame algorithm by NF. And we use like a Hamiltonian based unfolding process over the angular coordinates to unfold into a secondary structure. So one of the perks of this angular representation is that it maintains the connectedness between the individual consecutive residues, because it is based on certain bond angles between the certain residues. So it maintains like a backbone structure. It implicitly induces a certain backbone structure already, because it does not depend independently on each of the residues. Whereas this frame representation is independent of each residue. It doesn't define the connectedness, but the angular representation defines the connectedness. That's why it makes it more easier to preserve the topological or structural integrity. So we utilize a Hamiltonian unfolding process over the angular space, which is denoted as Z, which unfolds the protein into like this linear chain, also known as linear beta sheet in the biological terms, without like having collisions. So that's how it is formulated. It's a second order process. So Z of t, the temporal derivative depends on the velocity vt, and the temporal velocity depends on this unfolding potential, which we define as the u of Z of t. And there is a certain drag force, which is like a friction. So one of the things about the Hamiltonian dynamics is driven by always by an energy term that defines how the system will evolve in a certain energy potential, which is defined by this U. And this K, or the friction force, kind of modulates how things happen in real life, because they are not, when protein like structure is folding or these things happening, it's not happening, let's say, in vacuum. There is always a certain environmental things happening, or environmental friction coming into like, let's say, happening in cells or somewhere. There is certain liquids or like plasma happening, which kind of gives it like a certain force or those certain kind of things. So we define like an augmented potential. There is a target one and there is a repulsion one. So the target defines like the potential to our prior state. Let's say we have a target state, let's say a linear chain of amino acid is defined by the Z of I of target, and there's the harmonic potential between the current state and this. So this describes like a driving force to the, let's say, the linear strand of amino acid chain. And then there is a repulsion force. So this repulsion force is based on the Coulomb repulsion potential, which defines like how, which makes sure that during the forward process, your point residue shouldn't collide into each other. So this makes sure there is a repulsion between them. And we define, using this process, we define like a target distribution, which is both for the position and the velocity, since we are modeling a second order system. So the velocity, we always assume is from a certain Gaussian distribution, which is easily there. And in the position one, we assume it's at a certain, let's say, u beta and sigma beta. So the mu beta represents like the certain angular representation for this kind of linear chain of amino acids. So there is a certain angles that characterize this linear chain of beta sheet. That's what we use as like a target distribution, but it is flexible. One can use like any kind of representation or like any kind of angles as like a prior distribution. So we chose it to be just to be like more, let's say, having a like a physical prior. And we know that since we are defining a flow in the angular space, this also corresponds to a flow in the Cartesian space because they are bijective in nature. So we can use both of the representations to train our model on and to give like a both views to our model. Since there have been like much more development in using IPA, invariant point addition, those kind of methods in Cartesian domains, which are missing on angular domains. So they represent like it's much more flexible to model in the Cartesian domain as well. So how does it work? So like let's say recapping. So this is like a general SE3 protein generative flow looks like. So they have like a structure. They do this SO3 flow and R3 flow, go to random noise. So how do we fit in? So we just we kind of replace uh, the R3 flow on Cartesian coordinates to like our unfolding flow because we are only concerned with like let's say the translation. We can still have the rotations kind of pretty randomly because they kind of doesn't tell that much, like let's say because when you are moving towards like let's say a linear chain, we only are concerned with the like the R3 position of the C alpha where it will be going. But the rotation is just like an alignment of like how does it like looks like in like um yeah. And it also doesn't correspond to like because all the terms that we are trying to enforce, like let's say in the target and let's say in like a repulsion term, only depends on the R3 Cartesian coordinates and not in the rotational space in those. So we can directly plug it in here at SC3 flow and we have it will take to like a linear beta sheet prior and we try to learn the reverse. So we utilize like a multimodal neural network architecture to model both sequence and structure information because we want to incorporate sequence information because from a certain linear strand of like let's say linear chain of amino acids there can exist like multiple different protein structures from there. So we use like a pre-trained ESM2 language model for a sequence encoding, and the structure encoding are just IPA transformers using the AlphaFold from the AlphaFold2 architecture. It's also given in this plot diagram. We have like the noise states like XT, RT, the T, and S, and the sequence encoder that is passed through the combiner module that concatenates and adds or like merges these two representations into together. And then at the final, there is a decoder. So it's like an encoder decoder framework that gives you the corresponding velocities, like because we are learning kind of like the velocity and the any auxiliary outputs. So we utilize flow matching to train our method. So it has been seen like if you define like a certain Hamiltonian dynamics, you only need to learn the velocity in the reverse direction. You don't need to simulate the second order dynamics from reverse. So we can, and you can just directly train by fitting the velocity rather than using any other thing. So conditional flow matching works perfectly well. It has been proven. So we utilize conditional flow matching to train our method. We have two losses, the SO3 loss and the R3 loss. This velocity is given by our unfolding process. The V V of xt given x1, x0 from the former simulation. And this velocity is by the simple SO3 flow. In order to learn more, so one of the difference that we have with respect to the linear diffusion flow, that our trajectories are not linear in nature because we are following a certain potential. It is non-linear in nature. So we need more penalty, or like say we need to have like more penalty terms so that model learns these kind of non-linear trajectories. So in order to do that, we introduce like a look ahead loss. So what does it do? At each time step t, it kind of also predicts as an auxiliary output, like what will be the state at a certain, let's say like after like a certain delta t from the t, also defined as an s. So like let's say you sample a certain t between like 0 and 1. So the model tries to predict like for a certain s which is between t to 1. So it always tries to predict like what is the state after like let's say 5 steps ahead of the current time, or what is the state after 6 steps ahead in the current time. And then we introduce like a certain penalty term in also for that, so that the model knows it's like more like a non-linear rather than a linear term. And also we have like more losses, just utilizing from the baseline as well, like the histogram loss and the terminal state loss, just to make them more better. Now going towards the experimental evaluation, so we train our model by the filter structure from PDB. So we have monomers, which are single chains, having the length distribution between 60 to 512, also represented in this histogram, which has like a little bit abnormal peak around 130 or 40. And also they have high secondary structure content, less than 50% loops. So we get like 24,003 structures from current PDB archive. So we also do, so one of the tasks that we do is unconditional generation. So we train on the whole data set and just generate unconditionally protein structures. And then the other one is a conditional generation, that we want to have like a certain sequence and then generate as a certain protein, and then we compare it to the real one. So in order to evaluate that, we try to have like a certain training and test set, so that we can train our model on the training set and evaluate it on test set, such that we can evaluate fairly and like goodly, so that there is no data leakage between the training and the test set. So we split it the total 24,003 structures based on clustering them on 40% sequence similarities and splitting the clusters. So like, let's say we take like 100 clusters, there are total 120 clusters. So we took the 100 random clusters for the training set and the 20 different clusters for the test set to evaluate our method on. And this is the training and the test distribution of how does the look like with respect to lengthwise, and they're pretty kind of similar. Showcasing, okay, we have like a good evaluation capability in the test set as well, starting from lower range amino acid like 60, and we have until like 500 length residues as well for the evaluation. So these are some results for the unconditional generation. We compare to all the current state-of-the-art baselines like RF diffusion, fold flow, Proteina, frame flow, fold flow. Our model achieves better results than the models that have been trained on the same data set. So usually RF diffusion, Proteina, fold flow, they are not trained on the same data set. They're trained on much larger, let's say 20 million on AFDB data set, combining all the external synthetic sources. So the direct to direct comparison is not kind of fair. We also have like the methods that have been trained on the same data set, like Genie, frame diff, frame flow, fold flow, and phase flow, and recu. These are the certain methods that have been trained on the same PDB data set, offering a more fair apples to apple fair comparison. And we see like it performs kind of really well in learning and having good state of the art results, let's say. And here are some of the proteins that have been generated from it. In order to move into the conditional generation, so this is like the one on the sequence condition generation test. So we had like a test set of sequences that we wanted to generate our protein structure on. So we utilize this test set. So apart from ESM fold, so ESM fold is trained on like say 65 million uniprot data set thing. So it's kind of, you just put it there like for as the gold standard of how they are. And we also retrained these methods, which are co-design methods. It's a multi-flow, fold flow to our method on the training data set that we had, and evaluate on the test data set to see in a head-to-head apples to apple comparison to see which methodology has really good performance rather than scaling the data. And we observed that we are able to get a little bit better performance. And we also try to extend it to this Cameo 22 data set, which has been used for like all the folding methods, such as eigenfold, ESM fold, alpha fold. Obviously, it will not be that good as they have been trained on much larger data set. But we want to highlight that when we compare with fold flow 2 or any other these method, the amount of CA clash rate, which is one of the key factors determining if the protein structure is physically viable or not, is really good in our method, is only like 0.2 or very less, which is really better than even like eigenfold or like, let's say, open fold in them. And also have like the really less peptide bond break as compared to the percentage with respect to like ESM fold and this, because we are kind of trying really to model this kind of like trying to avoid the structural collision and trying to model the structural integrity. And these inducted biases have made such that we have really good, very less clashing rate or like peptide bond breaking rate as compared to the other methods, which just directly use uh, 3D random noise as their prior. And lastly, we have one motif scaffolding experiment where we have a certain motif as like a conditional input, and we want to generate like the whole protein around it. And we compare it to like standard methods like frame flow, RF diffusion, Proteina. Here also the Proteina, Genie 2, and those are trained on much larger data sets, so we can't have fair-to-fair comparison. But we found out that our method is able to beat even Genie 2 and RF diffusion in having more successes and task as a best method. So finally going, and these are some visualizations of how does it actually really look like in practice. of the inference trajectory of how the protein starts from like a linear chain and kind of folds it into like a certain protein structure generated by the model. Yeah. And finally, that's a wrap. So we have tried to devise like a physics-informed noising process for protein structures for now, and we combine it with SE3 flow matching and try to learn the distribution of these protein structures. And what you will find more in the paper, which I have not discussed, like how this unfolding process is SE3 equivariant or invariant with respect to rotations and translations, just to make sure that we are abiding by the symmetries of the nature. And also how do we parameterize our model, neural network architecture, and also an ablation studies over like what is the inference time, trainable parameters, how does the energy weightings affect the forward process. As we see, there is a certain energy that needs to be weighted in the K1 and K2 weights, and also what is the effect of these time steps in generating the protein structure. And that's all. Thank you for listening.
Thank you so much for your presentation. Very interesting work. Um, so I have a lot of questions, but
Yeah.
there are a few audiences here. So if you have any questions, you can
Mm.
ask using the Q&A button on the bottom, or you can also post your question on Slack. So feel free to do so. Um, in the meantime, I was wondering, so I don't have much background in protein folding to begin with. So I wish to start with super basic questions. Um, in practice, can all protein, like you said, monomer, right? So can all
Mhm.
monomers be uh, kind of unfolded into a single strand, like a straight line?
Well, actually, that's the one of the biggest question the physicists and the biologists still work on. So obviously, it's kind of like they. Okay, so I think it's like they can be obviously like if we minimize like what kind of forces that we use. So usually the process of unfolding folded protein structure involves many things. We have not included like most of the things. So there are like certain thermodynamical conditions, energy barriers, and certain kind of things that we need to overcome, which are still kind of being understudied and these things. So but here we obviously like most of the methods and they also don't incorporate this like certain kind of thermodynamical condition, environmental condition, and certain kind of things. What is like because those affect the structure of like the protein and how does it work. But here we were trying to like incorporate the certain minimal inductive biases that are present there. Let's say, like if a protein chain is colliding with another protein chain and your model generates it, there is no way possible that it can exist in real world life. Even in like any thermodynamical condition or any certain kind of thing, there cannot be any broken, like let's say the chain structure and those things. Those cannot exist in real world life. So those were like the minimal inductive biases that we were trying to incorporate in it. Obviously, by extending it to having thermodynamical conditions, and those are really good step, but there is also a little bit scarcity on the data side because we don't have that kind of a data, so that limits like our ability to model those methods as well, because we can easily like extend it to like learning, like say the conformational temperature based dynamics by just modeling the like say like the prior distribution with let's say a Boltzmann coefficient with like exponential to the like kbt, because those are kind of used for like the temperature based scaling to have like at a certain temperature certain will be more preferred, and at a higher temperature all will be like more equally probable. So those are like kind of strategies one can also incorporate in that.
Mhm. Makes sense. Thanks. Um, Sounds good. I was also wondering about the another basic question. So when a protein is folded in practice, in reality, they always start with a single kind of strand as well, right? And there's no exception at all.
Yeah, actually, yeah. So yeah, yeah, actually, this is like a very because it depends on protein to protein, like what kind of protein you are modeling. So like let's say for example in the cells, how does it happen? So what happens like because an mRNA goes into like your ribosome. So ribosomes are the like the cell organelles that make protein. So what they do, they go sequentially one by one, and then they create like a linear, let's say a strand of like the protein comes out, and then it goes into the chaperones. This is like a special kind of like a structure like present in the organelles. It goes into the chaperones, kind of like in a wavy form, not folded, And then in the chaperone, something magical happens. Let's assume that. And then we come out like a folded protein. That's actually it kind of happens like that. Not going into like how does it happen inside the chaperones, because they have like a certain kind of like say apparatus or structure that modulates this kind of like a folding kind of behavior. And also like there is a certain dependence on like what environment the protein is in, that how does it fold, because
Mhm.
certain conformational like fold switch will be more preferable at a certain temperature or at a certain environment rather than the other one.
Mhm.
Yeah.
Yeah, thanks a lot for the answer. That really clears up mind for the mindset for listening to the
Mm.
materials here. Um, Now, regarding the evaluation metrics and the and overall motivation for generating protein structures, I was wondering if the evaluation metrics, like I forgot the names of these metrics, but if these are kind of inspired by some practical needs. If so, then what are potential practical application scenarios for protein structure generation?
So, I don't know why it is. Okay, let me show. I
Design,
think it's
yeah,
going. Yeah, it's going back. Okay. Yeah. So, what?
yes,
So this
design
is
approach.
like,
Oh,
yeah. Right, this is very good. Can you still? Oh, still do not showing it. Wait.
it used to work, so I hope it
Yeah.
works.
Yeah, so I think these are like the standard kind of metrics that have been used, but they are not like foolproof metrics, I would say. So in
Hmm.
the designability, this is like saying, let's say you have like a certain protein structure, you use like a certain, let's say, protein MPN or a certain kind of an oracle to tell what sequence it can be. So like the inverse design problem that we have, like given a structure, tell me what sequence can it fold into. So
Hmm.
you have like a bunch of sequences. Now you use certain, like say, alpha fold or ESM fold to get a structure out of it. And then you match these two structures, like the generated one and the predicted one from ESM fold. And then if the amount of structure, the fraction, like the amount of structure, the RMSD is less than 2 angstrom, it resembles, okay, it's like nearly similar. And that's how they characterize this designability as a metric. Although it is limited to what your protein MPN and ESM fold can model. So it doesn't mean like if your model generates like let's say a different kind of protein structure, which is perfectly valid, nothing collision, it cannot exist in real life. Because it's just not the ability, because there is a certain oracle that is limited in generating those sequences that can fold into that, let's say, the structure. So this is kind of like this oracle, the metric is limited by the expressivity of the oracle that we are using, because we are using ESM fold and protein MPNN in between to generate, like, let's say, this designability criteria. And also this novelty and diversity just measures like how good the protein structure is in, like, let's say, compared to like the proteins present in the PDB structure, or like how diverse it is. Diversity is measured based on like also like in terms of secondary structure content, like does your model have more alpha helix or beta sheets. It has been also shown that usually alpha helices are more designable than beta sheets. Usually they are just occur like more in nature, and all the protein models, like genetic models, are kind of more biased towards generating alpha helices rather than beta sheets. I think you can also see it in from the secondary structure content percentage. So they're always like, if you see like they're more towards this. It's also like one of the factors, because we always want to try to maximize on a certain metric. So we know that alpha helices are more designable. So we have a bias towards alpha helices, the model will be more designable. But it doesn't mean it can generate like all the coils and those kind of stuff very accurately. So these are kind of like, let's say, having like these kind of trade-offs there for these models.
And okay, thanks a lot for the explanations. Did you say novelty is all? What is novelty measuring again? Is that novelty against the training data?
Yeah, so novelty is measured across the training data. So what they do,
Mm.
this is like a TM score. So this is like a template matching score. So they it's kind of like a method, or that's a matching score that matches between two protein structure of how similar they are.
Mhm.
And then we rate like how many of the TM average max TM similarity we have based on. So the more lower, the more better it is, because then it will be less closer. So we see like it's around like 45% similar or 40% similar, around like those values. Like that's the maximum value that we can get. But there can be much more lower. And also like the TM fraction is like more, let's say they take like a fraction of the each
Mhm.
residue length based thing, and then they compare. It's like a little bit more in detail in this. But they use like this kind of TM matching thing, because that kind of tells you like how much close your a certain protein is to the whole data set. And then you compute like an average of it to just tell like what metric it will be really good.
Makes sense. And finally, the reason why you're measuring all these metrics in this kind of community is because eventually you want to, for example, for designability, you probably want to kind of synthesize such a protein eventually after your generation. Is that the main reason why you?
Yeah, well, yeah, that's actually a very, very interesting question. And the thing is, so what in my like, I think is like the designability does not correlate with synthesizability actually. There has
Mm,
been like no study like telling like, okay, if I have like 100% designable protein, it doesn't mean that it can be synthesized easily, because synthesis is like a completely different thing than a protein structure or this thing. Because synthesis comes from like, let's say you are able to have like a, because that requires like a much more like, let's say these kind of
mm.
like methodologies which have been used in like biology, like let's say I'm not able to recall like DNA proliferation, like making custom RNA such that ribosome can do it, temperature conditions and those kind of stuff, which are not being modeled at all in like all the methods. So it doesn't, I would not say like if it is a designable, it can be synthesized, but what we want to aim is obviously want to have like more designable protein structures by this metric. So it can be more closer to what we have. And it's kind of like having a metric to evaluate different methods. So it's kind of like, okay, we have more designable proteins. Okay, it's much better method than the other one. So we can use it for various other things. I think there are like other metrics, I think as well. In like the conditional generation, it makes more sense because we are given like a sequence and then we evaluate those structure that have been there. So one of them is always like the root mean square distance, like how close they are in the structure. And also these are the methods that have been used on like this Cameo 2022, like the TM score, as I said, like the matching between the structures of the two protein structure, that will be the true and the generated one. So this gives you like a template matching score, the RMSD, and these are all like this IDDT and GDTTS, certain like biological metrics to showcase like if they are higher, the better the protein is able to, you're getting much closer to the ground truth. So these are usually, so they are always like based in like a predictive modeling setting to like check like if it is able to model a ground truth or not. There is no as such relation between them and synthesizability, because synthesizability comes with its own set of problems that are there.
Okay, okay, so that clarifies the metrics. But please explain to me, like, once you build a very good generative
Mm.
model for protein structures, how are you going to use that?
So usually it is used in like different scenarios. So one of them is like also like let's say motif scaffolding. So this is like one of the things. So what they do, like let's say you know like a certain active binding site, like let's say consider enzymes because we can just extrapolate it. You have a certain active binding site of an enzyme called thiozyme or certain thing. Now you want to generate like a certain full enzyme structure, what can bind to that. So that is like a condition on those, like let's say binding site. I can generate the whole protein structure, or like what can be existing protein structure that I can make, or like those kind of stuff. Mostly it is used in those like binder design or like those kind of stuff. So one of the yeah, that's like one of the ways. And the second one is obviously like to have like much better protein folding methods or like those kind of stuff that generalizes very well and those kind of things. That is also like one of the things to check. And also like in the real world, yeah, I think it's just like because since it works on proteins and so it can be extended to antibodies and those kind of stuff. Because in antibodies you only know like a certain CDR sequence, and it has been on less data set because we have less antibody data set as compared to the protein data set. So we want to try to learn, let's say if I like let's say train a foundational bake model on protein data set and I can fine tune it on antibodies data set, can I get like much more information and much more better performance in generating new antibodies and their structure as well. And also like this motif scaffolding in based on antibodies, so that I can find with respect to an antigen what antibody structures I can generate. So these are like one of the, some potential those, and also like protein binding affinity and those that can be easily built in those methods. I think many of the AlphaFold, and I think they do kind of the same things.
Mm. Yeah, thanks a lot for the expert's explanation on this matter. I also believe synthetic data will continue to be more important, right, for training foundation
Yeah.
model in a lot of domains. Okay, and um, going a little bit more kind of futuristic and a little bit more general, can you explain to me, maybe first of all, do you recommend people after you, like students who are starting their PhD, to work in this domain, or should they work on something else?
Oh, that's a very hard question and interesting. Well, I think it's I think it depends. I think it first of all it depends on the interest of the student or any person that if he's interested in doing this like this for proteins and those things. Obviously, then if he's interested, then I'm pretty sure he should do it. It's not
Mhm.
like also like I would say like don't get like let's say disheartened by the amount of papers coming or like those things. I think
Yeah.
it's like you need to find out like a certain problem and then try to come like in a good way to solve it or like in a certain way. Yeah, I think there are still many problems or like the challenges there in the these kind of like tech, like the structural biology or those kind of domains that biology people know more, but the people working in computer science are less aware of. Because if you go to like let's say biologist lab and ask him or her like what are the things that you need and how do you evaluate your alpha fold or like these kind of methods, then you get like more insight on really how do it actually works.
Mm.
Well, yeah. And rather than like, because we are not, so one of the things, like as you mentioned about the synthesizability and those kind of stuff that has never been incorporated in these kind of domains as well, like in the protein structure or these kind of things. So one of the domains like which is similar also to this, like this retrosynthesis, like to tell, like let's say to synthesize a certain compounds, you need to have two or three more reactants that
Mhm.
combines and you generate a certain compound. But that's not how it goes in real life. You have like a certain environmental condition set, you have a certain let's say solvent, you have a certain temperature, you have certain extra conditions that is never being modeled in like say machine learning models. So it kind of there is a gap, like you cannot just
Mm.
directly use it, and it most probably gives the worst results because your doesn't know how to incorporate those temperatures or those kind of stuff. So those like
Mm.
one of the like things that people are trying to like come up about, and so there are like still many of the challenges around enzymes and proteins and other ones. I think it will still be there a lot.
Mhm. Interesting. Can you also share your thoughts on the interplay between academia and industry in this community? Do you think they are kind of sharing, tackling a similar kind of problem, perhaps with industry kind of beating any of the approaches from the academia, as in most other domains of machine learning? Or do you think they are kind of sharing, like partitioning the space in a efficient way
Mm.
on different problems?
Yeah. I think well that's a very interesting and more harder question to answer because I'm not from industry.
Mm.
So I think it's like always because I think the industry people might be a little bit more above, because I think due to the one reason that they can have access to certain proprietary data that we don't have or like in academia doesn't have. Like they have the data, they have conducted tests and those like wet labs and those things, which the
Mhm.
academia doesn't have. Like we cannot have. That's not possible to have. So this obviously like kind of limits like what we can model or what we cannot model. And
Mhm.
also like there is a certain, let's say, so this is coming to like let's say one of the conceptual like kind of difference or like this thing that they have, and then there is always like this technical differences that we have because they have let's say access to a lot of GPUs or whatever things, and so they can always like use a lot of resources. So that is also like one of the key difference. But I think like there is also like more flexibility in academia or like these things to work on different problems as compared to industry, because industry are more focused toward like let's say a certain product or like this thing at the end of the goal. But academia is like more like exploring, like a curiosity based exploration or trying new things. So that, because sometimes like okay, a curiosity based exploration can lead to much better results or like let's say things rather than what customly
Mhm.
people will do in like just focusing on one thing. So it's kind of like a balancing out, but yeah, it's kind of like a symbiotic relationship, I would say. Like
Mm.
without one, the other one would not kind of be aimless, I would say, because there
Yeah.
can be like certain problems they will tell, and then okay, we'll try to work on it and see how does it like develop this field further into that.
Mhm.
So that's kind of like a going like a symbiotic relationship.
Hmm. Yep, thanks a lot. And um, maybe one last question from my side is promising future topics for this topic, right? So I think you mentioned a few of them, like from some practical point of view.
Mm.
Um, I wonder if you have any other ideas from practical point of view, as well as perhaps from a conceptual theoretical point of view, like your work has contributed, the work that you presented now.
Like in conceptual ideas about like what the downstream applications or like in what
Mm,
or like in a
what I meant is,
new.
in this work, you presented something that is
Mhm.
more kind of, I would say, a little bit general technique as well, where
Mm.
the problem could be formulated as generative model, where you have some physical constraints, which are perhaps
Mm.
represented using some dynamical systems. So
Mm.
I think that's very kind of general
Mm.
technique which can be applied in many other domains. I
Mhm.
wonder if you have further ideas or if you feel like there's a big gap here to fill in and so on, in a similar sense.
Yeah. I think it kind of okay. So I think this whole thing, like incorporating physical constraints into the dynamical systems or those kind of things, kind of resides on the gray area of, like, let's say, incorporating physical biases into this machine learning architectures, which have been not been only limited, let's say, proteins, but have been explored elsewhere around, like say in weather, climate, and those kind of stuff. And it has been proven to be really good as compared to simple machine learning, let's say, applications. So coming from like let's say like if you go to like a scientist in like natural science and this that person would always be thinking like why can't you just use these kind of Newton laws and those kind of things, incorporate them into your machine learning models. But there is always like a kind of like what the say like in a bitter lesson, let's say when we try to incorporate this or we are trying to scale with the computation, either them or will work kind of fine and we'll get the same amount of performance. So it's up to like people to like, do we want to really incorporate those or not? So that's like one of the things. But I think like more in like say biological or those things, because we know that the data is scarce in these kind of fields still now, because we don't have that much wet lab data, synthesizability data, or those kind of data in those sense. Synthetic data is obviously there, that I know, but those are like also in limited in a certain sense. Like let's say you have synthetic protein structures, but you don't know like if they are synthesizable or not. You don't know what temperature do they exist, the conformational whole dynamics and those kind of stuff. So that also like kind of like bulges in, like when you want to really make like a certain model for that the scientist or this like real world biologist or chemist or like say any of the other people kind of will use. So those are like still kind of like let's say more like a deployment kind of also a problem. Like let's say it's not like a fully let's say research. It's kind of like a research engineering deployment problem that they are existing. But also I think methods will also be like developed based on incorporating those. Like let's say you have certain missing data in your observe some missing data from like a certain place. Now you need to do inference or like certain things on this missing data. How do you do it? Because in real world you might not have access to the whole data. So those kind of things are always around the corner. And also like one of the things is like generalization to outliers and those things, because there is always there can also be like certain sequences or protein structures that are completely different from what we have observed and they are still very good, but your model will not be able to model because it's a one or two samples from outside, very very long tail distribution. So modeling those things are also like kind of like very important, because we only want like certain only some specific proteins that we know that are really good. So we want to maximize the precision, not the recall in those kind of sense. So that's like kind of like methodologies I think people are still working on or like still going on in like at least in the computational biology for like these things. And also like many other things are kind of coming up like RNAs and those kind of things which haven't not been explored, but currently now they are being trying to build it up because we have AlphaFold and those things for proteins, but RNAs doesn't work like proteins. They are completely different things. So because and then there are also vaccines in RNAs, like there is an mRNA vaccine for COVID and Nipah and certain kind of thing. So we want to also use those, like model those things as well, which are much harder to model, and we have much less data for them. Like if you see, like let's say RNA PDB, they have only like let's say 10,000 structure or maybe less, but the protein PDB and AFDB has so many structures that you can use to train model. But in RNAs or those kind of domains, we have much less data. So we need to see like how we can also utilize what we have done so far in, let's say, in proteins and stuff, and translate it to like in, let's say, in RNAs and this, because I'm not sure like if simple repurposing would might really help, or we need like something completely like new to solve that problem in those domains, because kind of they have different complex systems or modeling biological systems. They're completely different.
Oh yeah, thanks a lot for a very insightful answer. Um, can I ask one last question from my personal interest as well?
Yeah,
Uh,
yeah, sure, 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 sure,
the
sure.
So in the other domain, so in the domains that I'm working on, for example, we do look into generative models as well, but we focus on the concept called compositional generalization, which means, yeah, you have seen certain kind of
Mhm.
factors before in different contexts, right? But this particular combination of the factors I have never seen before, or or in generative models, the question is whether you can generate something with the known components in a different composition.
Mhm.
Do you think this question also, like, there's an analog of this question in your domain as well?
Hmm. I think there. Hmm. I think there obviously there is like in, let's say, in proteins and stuff, but I always like see, because I think in like the when you do like compositional generalization and like those kind of stuff in like say in images and those, I don't know like if it can be really like I know it can be translated very directly, but there are like certain, let's say, because in images and like in those contexts is very easy, I would say, like let's say a cat or like thing, but like let's say you consider like say a protein or like let's say a molecule, you are having like certain functional mappings based on the structure and the amino acid
Mm,
types. So like let's say if you want to have like a complex functional mapping, then it will really mess it up. It can have like a very, like let's say, not a easy way to find, like having like a composition would suffer like do it. If you like let's say if you take like a mixture of experts or product of experts in those, does like the mixture of expert and product of expert is able to model like let's say the whole functional composition that we have.
mhm,
So it kind of always like tinkers on in between, like okay, up to a certain point you can model it like in the certain sense, but then is always limited about like how you are composing them together.
mhm,
Like what is
mhm,
the good way of composing, let's say, two generative models. Like say if you consider like say protein structure, like one of them
mm,
gives you a really good, let's say, hydrophobic proteins, and one gives you like much more synthesizable proteins. Now I
mm,
want like a certain protein that is more also hydrophobic and synthesizable. So
mm,
does it really work if I just compose them, or does it have, because it's not like a kind of a linear relationship between the hydrophobicity and the protein
exactly.
structure. It's kind of very non-complex functional mapping, which is,
Mhm,
you cannot evaluate it in like, because it's like a oracle. You need to have like a wet lab test first of all. So you need to have like a certain oracle to test it then. So now you're biased by the oracle. So that's like kind of like a little bit still an area of exploration, I would say. But it's kind of like, yeah, it's kind of limited to like very simpler systems, I would also say. Not like exploring like say protein, because it's also like one of the things, because we don't have the data of those kind
mhm,
of things. That is also like one of the things to like how do we do it, because there is also
mm,
like functional design of de novo protein
mm,
molecule protein structures haven't been like kind of done, like in sense, like let's say I give you like certain properties, like let's
mm,
say I need this. I'm pretty sure many of the like these methods are not made for like
mm,
those kind of things, because we don't, because if you go like let's say in PDB or AFDB, the functional properties are not that much there. There is not that
mm,
much data on like those things. There's only like okay, the protein binds here and then done. This binds to that, and like yeah, binding structure is there and stuff, but not like that much functional thing. It's more in
mm,
like molecules that it can be really done in that way.
I see. Yeah. Thanks a lot for the insight. So, for this domain, maybe the total amount of data is the problem still, right?
Yeah.
For web data, we have a lot of data, like from the web and all those
Hmm.
images. So the models have seen definitely each of the concept at least once.
Mhm.
But the question now is whether they can be creative. But I guess we are speaking about different kind of regime of amount of data.
Mm,
And
yeah.
also like there's lack of annotations, I guess, for each factor in the, and it's also not clear. Like image is super localizable. Like each object has their assigned pixels,
Yeah.
whereas I guess that's not as easy as
The
in.
case in protein.
Mm,
Yeah. Yeah, I think like there is also like nowadays people are doing the structural compositionalization and stuff. Like, so for example, let's say I want to have like a certain alpha helix in a certain place. So you have like,
mhm,
okay, let's generate a beta sheet or like this thing. But it kind of
mm,
also resonates with like this kind of painting and those kind of stuff. So
mhm,
like you
mhm,
have like let's say I want a alpha helix, but then I want like the rest of the protein to be hydrophobic.
mhm,
Like this is a localized composition that is happening between the structure and the functional. I fix like a certain part of structure, and then I want to generate the other part of structure, making sure that will be hydrophobic, or like will
mhm,
have like a certain property. So those kind of compositionalities also kind of like work in this kind of like domains,
mhm,
which is kind of like a little bit different than images. Like say I make like a half cat image, and then I want a half dog below it. So it's like, okay, I gave
mhm.
you a half cat image, now I want a half dog or half peacock below it. So those kind of like things happen in like this structural and those kind of things. So it's kind of a little bit more complex, but also like I think it's like interesting to explore in those domains to see like how we can really incorporate those and extend those things into like when applying to like real world complex systems. That is
Mhm.
always like a certain challenge of applying this machine learning methods there.
Right. Thanks a lot for giving a very deep insight into this domain and making comparisons and giving structure to all these chaotic
Mm,
questions. Very nice. So I think there's no other question from the audience so far.
mm.
But I also wish to mention that Yogesh, are you on Slack channels
Yeah,
for?
yeah, yeah. That's all.
Okay, if that's the case, then we have a possibility to continue the QA on Slack channel
Mm,
in the future. And also there's YouTube video to be uploaded based on the chat we had today. So yeah, I guess people can reach you out, or Yogesh,
mm.
with follow-up questions in the future.
Yep, sure.
I wish to thank you again for presenting your
Mm.
work and engaging in very interesting discussion today. Um, and thank everyone who joined the session to listen to the discussion. Okay, so thank you so much, and